Welcome again to this crash course in muscular dystrophy. Every time you smile, you take a step or hug someone, your muscles are at work. You got tiny little fibers, millions of them, all working together in harmony. But imagine for a second that this stops working, that they no longer cooperate together. And hence, even the simplest task become complex. And this is the sort of daily challenge faced by many individuals with muscular dystrophy, a condition or group of conditions, I should say, quietly robbing independence and strength and those tasks that we often give for granted and sometimes life itself. So muscular dystrophy was described, I should say, about 200 years ago, and yet scientists and clinicians, doctors are still puzzled by a lot of mysteries around this condition, and it remains incurable to date. Today, I'm going to tell you a story then about science, research, resilience, and genetics, and cutting-edge innovations that we're working on to try to solve some of those mysteries and hopefully help the development of future treatments for this condition. So I have the privilege to work across these three amazing institutions, the Crick, UCL, and Great Ormond Street Hospital, and my lab is focused around studying and modeling human muscle development and degeneration and regeneration in severe neuromuscular disorders of childhood, mainly in muscular dystrophies, to develop future treatments for those. And we work really at the crossroads between those three large areas of science and technologies, stem cell biology, tissue engineering, and disease modeling. So back on to the topic of today. So what is muscular dystrophy? What is it that makes a muscle looking like the one at the top? We're looking at, in this cartoon, at a muscle from the top, if you wish, in a so-called transverse section. What is it that makes that muscle turn into a tissue, another muscle which contains a lot of scar and doesn't work properly, like the one you see at the bottom? So a few words about muscle first. As you might know, it is the most abundant tissue of our body. It is the main output of our brain, and it has a pretty characteristic structure. I mentioned already to you those little muscle fibers. This is a cartoon representing one of those, and you can see that they have they look like long tubes, if you wish, with a lot of small nuclei. The nuclei is where the DNA is stored on our cells, where the genes and the information is stored. And at variance with most of our cells in our body, which contain one nucleus each, a muscle fiber contains hundreds, sometimes thousands of nuclei. And this is already a particular thing that they have. They live wrapped around a layer of so-called connective tissue that protects them and helps some of their functions. And they are in close contact with a number of other important structures of our body, like the blood vessels or the nerves, which guarantee the transmission of the stimulus to the muscle fibers. And they also contain a population of stem cells next to them, called satellite cells, which help regenerating when there is some damage. Muscle has multiple functions, like movement, posture, regulating temperature, maintaining protecting soft tissues, and so on and so forth. Now, I just would like to wrap up and summarize what I just told you in this very nice video produced by one of my former mentors, Shargim Tashbash at the Institut Pasteur. Now, you see a cartoon of our quadricep muscle, and now we're zooming in into the muscle fibers that I mentioned to you before, and you see that the fiber, now if we strip away the connective tissue, so the top layer will highlight the presence underneath them of these little green cells, which are the muscle stem cells. You've seen before already the blood vessels and the nerves next to them. And now if we open up the membrane of the muscle fiber, you will see the nuclei inside. Those orange spherical structures containing the genes in our DNA, next to those T-tubules, which help moving the salts around to guarantee the muscle contraction. These are the mitochondria in blue, the powerhouse of the cells. And this is the engine, the actin and myosin fibers, which help the muscles move. So then what happens when this machinery is not working properly? Well, quite often, unfortunately, this results into a muscular dystrophy, and those are primary degenerative disorders of muscle. What does this mean? So this is a group of disorders which primarily affect genetically, as in genes, which are involved in some critical functions of the muscle fiber. And there have been more than 50 genes linked to muscular dystrophies to date. So the problem is in the muscle fiber in the first place, is not somewhere else and then causing trouble to the muscle fiber. These conditions are progressive, so they get worse over time, and they result into weakness and wasting of the muscle. And if you were to do a muscle biopsy, as you will see later on, it is dystrophic, meaning that, as you will see, there are some peculiar changes that we can observe there. The conditions are, unfortunately, very often severe. The heart is also a muscle, so the heart is very often involved in those conditions, and our breathing muscles also are affected, so there is a lot of cardiorespiratory problems in those conditions, and unfortunately, invariably, all of them to date remain incurable. So I mentioned to you a muscle biopsy. Well, this is an example of a healthy-looking muscle. This is not a cartoon now, it's an actual slide, so we're looking under the microscope, and we're looking again at the muscle fibers now from the top, not sideways as you've seen in the cartoon before. And they're all very similar to each other, and this is how they should look. Now, on your right side, you will see a dystrophic example, where the muscle fibers are all different size already. You can appreciate that as a different thing from the one on the left. And in between the muscle fiber, you see that large amount of scar tissue, which obviously doesn't make the fibers work properly. There is presence of we call degeneration and regeneration, so fibers dying and others being made new, and also presence of fat tissue inside them. So imagine those fibers, now if we look at them sideways again, as if they were ropes, right, as in this example. Now in the left side, you see that each one of those ropes, so each one of those fibers is nice and healthy with a nice envelope, a nice sheet of the fibers. But the one on the right, they are quite thin, with different size, and some of them will be nearly gone, nearly broken. You can see the core exposed there, and at the next muscle contraction, that will probably snap. And this probably gives you an idea of what I mean by dystrophic already. Now, touch of history to understand where and when muscular dystrophy was discovered. I'd like you to bring you in this little village in the south of Italy, incidentally not far away from where I'm from, just a couple of hours drive, called now Pesco Sannita. Back in the days, in the early 1800s, was called Pesco La Mazza. And in this place, two brothers, the Caruso brothers, were born with a mysterious condition, and nobody really could understand what was the problem. Their muscles weren't working properly. Some muscles, like the calves, were getting bigger. They were experiencing heart problems, and their condition remained a mystery until they went to see the leading physicians of the time in Naples, and they got seen by this guy over here, who very few people know actually, Giovanni Semola, who was a clever boy back then, and started to look into this condition, understanding this is something new that wasn't really described properly before. And eventually, in 1829, he gives a talk into this place, which, again, I suppose very few of you will be familiar with, the Accademia Pontaniana, which was one of the oldest scientific societies of the world, actually, in Naples. He gives a talk there and presents this case of these two brothers with a condition which he called ipertrofia muscolare, muscle hypertrophy. And this will be clear in a while, in a few slides, why he called that muscle hypertrophy, as in muscles getting bigger. He gives a talk to this place, but he doesn't publish a paper. So a mistake we often do in scientists. Until a few years afterwards, these two other clever physicians, again in Naples, re-examined those two boys, those two brothers, Gaetano Conte and Luigi Gioia. Now they make a detailed description of the symptoms of the condition, which they end up calling, as you see there, scrofula of the muscular system. And it's not until the 1851 where eventually there is a very clear, unequivocal description by Edward Marion here in the UK of the pathology also of the condition, not only on how it looked from the outside. And he published a beautiful paper in which he shows in this very nice drawings what I told you already in the previous slides with the ropes, where you can see the fibers not looking healthy and some of them broken in those pictures. But you might have heard about Duchenne muscular dystrophy, and I haven't mentioned the name of Duchenne so far, right? Well, eventually in 1868, Monsieur Duchenne published the very first really comprehensive description of the condition in France in his seminal work that you see here. And he called it a pseudo-hypertrophic paralysis. Again, this term of hypertrophy, so muscle getting bigger, comes back. And why is that the case? Well, you see in one of his beautiful drawings of this paper, this is the first case drawn there, Joseph Sarrazin was the name of this boy. You see the calves, quite big. And for those of you with a clinical eye, you will also see the curvature of the spine, the lordotic posture, which is also pretty familiar, pretty typical in muscular dystrophies. Well, why those calves are getting bigger? And again, this will be clear in a couple of slides. Now, a few words about Duchenne muscular dystrophy then. This is the most common muscular dystrophy, and it's overall the most common primary muscle disorder in general of childhood. And it's caused by gene changes, by misspelling in the dystrophin gene, also called the DMD gene, which is a huge gene. It's 2.4 megabases, meaning 2.4 million letters. This is a massive amount of letters making a gene. It's the largest known on the planet. There is no other gene in nature on our planet larger than dystrophin discovered so far. And to give you an idea of the size, this gene lives on the X chromosome, so only boys are affected by these conditions for this reason. And the number of letters is pretty much the same number of letters of the entire trilogy of The Lord of the Rings. Now, why this condition is relatively common? Imagine copying over and over again by hand those books. Eventually you'll make some mistakes. Eventually you'll make typos. Eventually you'll misspell some of the words. And this is what's happening when the cells are dividing and are copying the dystrophin gene. And this is why it's also very difficult to anticipate and prevent as a condition. I told you that the gene is on the X chromosomes, and it's quite prevalent. Roughly 1 in 5,000 boys are born with this condition. And the gene makes a protein again called dystrophin, which effectively works as a shock absorber. So helps avoiding damage in our muscle fibers when they contract. The onset is usually in early childhood, and there is also a milder form of the condition called Becker muscular dystrophy, which has a slightly more benign onset later in life. There is variable involvement of the heart, as I mentioned before already, and of the brain, because dystrophin is also produced in those tissues. And again, unfortunately, it is incurable to date. So, but how lack of dystrophin causes a muscular dystrophy, right? So I told you that dystrophin is essentially the key function, not the only one, but the key function of dystrophin is that of a shock absorber, and essentially works as a spring in a trampoline, right? Where the trampoline is the muscle fiber and the spring is the dystrophin at the edge of the muscle fiber. Imagine jumping on a trampoline without a spring or with a rusty spring. You can imagine what the consequences are going to be. So the muscle fiber lacking the shock absorber is more prone to fragility. It dies over time, and it triggers a regeneration process. And this eventually causes continuous loss of muscle fibers, because the regenerated fibers contain the same genetic defect of the one which have just died. And this muscle loss is what the patients, the individuals experience as weakness. And the stem cells, which I mentioned to you before, get activated. They try to repair that problem, but eventually they get exhausted as well. Hence, the space left from the muscle fiber is filled by scar and by fat tissue. And this is why those scars in those boys were getting bigger from the outside, bigger with scar and fat tissue, but not bigger with actual muscle fibers. And this is why it was called pseudohypertrophy. And you can see that here. You see in blue in this staining the amount of scarring that is present in a Duchenne muscle compared to a healthy biopsy that you can see from on the top. So what to expect over time in a muscular dystrophy, and particularly in Duchenne muscular dystrophy? Well, at first the symptoms can be hard to spot. There might be delays in walking or in talking in some children. And some children have blood tests done incidentally, which end up highlighting some changes in muscle enzymes, or others have it done on purpose because there is a suspicion of the condition. Some children have challenges with learning and attention or social skills. I mentioned to you before that dystrophin is expressed in the brain as well. And over time, unfortunately, those muscles get weaker. And again, unfortunately, irrespective of how good they were doing when they're young, you see now in the early age, these are different colors highlighting different performances of the children over time in terms of losing the independent walking. By their late teens, which is the right side of the graph, the vast majority of them would have lost the ability to walk independently, unfortunately. Until a few decades ago, most of these children didn't even reach adulthood. Now the outlook is very much different with steroids and heart care, heart medications and expert medical care. Many young people reach adulthood and transition to adult care, but they still face major challenges and a reduced lifespan. And if you want to know more about what I just told you, please refer to this excellent, the website of the excellent charities that I mentioned to you. These are just some examples in the UK and all over the world. And we have also some representative of the Muscular Dystrophy UK team here today, if some of you have some questions for them at the end of the talk as well. Now, is DMD the most severe muscular dystrophy? This is a misconception that I often hear in scientific talks more often than not. It is the most common childhood muscular dystrophy, but it's not the most severe. And unfortunately, there are groups of muscular dystrophies called congenital muscular dystrophies, which are more severe than Duchenne. In those conditions, there is a very early onset, and you don't really need to be a pediatric neurologist to understand the challenges that these children face in terms of requiring respiratory support with their breathing, support with their feeding, and the muscles are extremely wasted and very much contracted in some of them. And unfortunately, they have a reduced lifespan due to the fact that the conditions remain incurable. And this is a group of conditions as well, not caused by a single gene, by multiple genes. Now, what options we have in terms of treatments? Now, I am focusing on this slide again on Duchenne, just because there are more options for that. We can broadly group the treatments in these three groups: drug therapy, as in pharmacological, as you see here, genetic therapies, and cell therapy, where we're actually providing an extra stem cell pool, which could help muscles regenerate, for example. So the broadest category of treatment options are those that are kind of non-specific, so limiting disease progression. They are usually anti-inflammatory or anti-scar, anti-fibrotic drugs. And I put here with an asterisk the one that are approved for clinical use in the UK. Corticosteroids, so steroids are the mainstay of therapy in Duchenne, helping delaying the loss of independent walking. And there are also very recently approved drugs like the one I highlighted here, vamorolone, and givinostat. Vamorolone is a modified steroid, if you wish, and givinostat, a new class of drugs which has just been conditionally approved a few weeks ago, I should say, almost. And then there is another broad group of strategies which are targeting the primary genetic defect. So this is where there is a lot of effort in the labs, but there are also some approved treatments for that. And stick with me for a minute or so now, because this is quite complex to explain. But essentially, what we and a number of other labs are trying to do when we are focusing on this approach is to provide either an extra copy of the dystrophin gene, and that will come in a second, or try to rescue whatever is left there in the cells that can be rescued and restored. And two examples are those that are put there. One is ataluren, which is approved in the UK, and the other one is the strategy of exon skipping, where we try to get rid of the bit of dystrophin which contains the misspelling and get a shorter but still functional dystrophin, kind of a shorter spring of that trampoline, if you wish. Those are approved in the United States. There are gene replacement strategies, as I said before. We take a modified virus to provide an extra copy of the gene, but the gene is so big that we have to shorten it. Again, make a shorter spring. Not completely functional, but supposed to be functional to some extent. And there are also more recent and cutting-edge strategies based on genome editing, like the CRISPR copy and paste or genomic surgery that we can do on some genes, and the cell therapy that I mentioned before, or alternative strategies, which are so-called compensatory strategies, as in we're trying to make the muscle fibers produce proteins which are similar to dystrophin, but they are not actually dystrophin. They are sort of sister protein of the dystrophin, like the one I said here, that I put here, utrophin. But if we have all these options, why are muscular dystrophies still incurable then? Well, there are a number of reasons. Some reasons are specific to the organ, if you wish, to the tissue, to the muscle, and to the condition. And I'm going to try to walk you through from inside out, from the DNA outside to the muscle fiber and to the muscle tissue in general. So if we go at the DNA level, I told you that there are several genes. So it's very difficult to have a one-size-fits-all approach, because we have multiple genes that have a multiple different mechanism causing the condition. And the DMD gene is, as my little son likes to say, ginormous. It's massive, so you cannot really squeeze it in a little virus and deliver it to all the multiple cells of our body. And then if we move to the cell, the muscle fibers are very peculiar. They have multiple nuclei. For the aficionados, I put some numbers there. In some cases we can have up to 100 nuclei per millimeter, and we have some muscles in our body which are over half a meter long. So make your calculations. You go above 40,000 per single cell. And those cells, those fibers, they become post-mitotic, meaning that once we reach adulthood, they don't grow and they don't proliferate anymore. So you need a cure that grows with the muscle, as I'll say in a minute, but it sticks there forever. And again, onto the muscle now. We're looking at the entire muscle mass. This is massive. It's about 40% of our body weight. You need to target all of those kilograms of tissue, ideally, including the stem cell compartment, which has its own challenges. The muscles are widespread across our body. You need to target the diaphragm. You need to have a delivery system which is good enough to reach all those districts, all those bits of our body. He has to grow over time. Think about the muscle mass of a baby and the one of a bodybuilder, right? Think about these two differences, and that treatment has to grow with those muscles. And muscles is a tricky tissue to work with in terms of immunity. It's highly immunogenic. Think where vaccinations are done. They are done intramuscularly in the muscles. And some of those challenges are also linked with the limitations and the challenges that we have faced in the therapies, both in the currently approved treatments, like the one I mentioned before, which unfortunately are not, life-changing. They just slow progression of the condition so far, most of them. And some of them, like in the case of steroids, unfortunately have quite a lot of side effects. For those of you familiar with steroids, you will know about diabetes, high blood pressure, increased body weight, and so on and so forth. And the new, the experimental treatments are also complex and challenging. And I put some snapshots on the right side of the screen to give you an idea of some of the challenges and side effects which we have unfortunately faced in recent years. Some of these genetic treatments have serious side effects on the liver, on the heart. They trigger a lot of immunity, and in some cases we've seen a lot of fatalities, besides the fact that they are remarkably expensive. So there is still some work to do to tweak that. And this work that needs to be done is often done with disease models which, in my opinion, are not optimal, as you will see later on. Current models of muscular dystrophy sometimes are not necessarily available. I told you that there are many different genes, many different forms of muscular dystrophies, and some of them don't have a good animal model. And when we're not relying on animal models, but we are relying on cells from patients, for example, from a muscle biopsy, where there are some bits of our body that cannot just be sampled. I told you that the muscle fibers need to be together with the nerve. You cannot just grow a nerve from a muscle biopsy in a dish in the lab, for example, or sample the heart easily. Fidelity. Well, there is the assumption very often done in the lab that a mouse is just a little human, and this is not the case. We've seen, I can talk for hours of examples where treatments have worked in mice, but they have not worked in humans in the case of muscular dystrophies. And the tissue, as I said before, is highly three-dimensional, right? And unfortunately, still today, we end up doing most of our discovery work, our research, into models which are, like you see in that little cartoon on the right-hand corner, a thin layer of cells in a culture flask, right? Very different from real life, in a way that I've got to make a joke, being Italian, that a lasagna, which is highly three-dimensional tissue, is not really a pizza. Both of them are good, obviously, but they are different, intrinsically different things, as you can see if you compare the two structures. Although people are claiming that you can combine them together. But now let's be serious again. Resolution is another challenge there. So we want a system that makes us study muscle development over time, and also to keep those cultures, those muscles in the lab, those cells in the lab for weeks ideally, so that we can study them over time. And there are ethical challenges. Animal welfare is a hot topic, and the biopsies are very often invasive, particularly in the case of the congenital muscular dystrophies that I mentioned to you before. Those are a neonatal onset. You just don't sample neonates easily, and you don't really want to do that very often, and sometimes you don't even need to do that to make a diagnosis, because you can make a diagnosis with a blood sample. And the costs are also quite relevant. The cost is an issue that affects also the therapies that I told you in the previous slide. Some of these genetic treatments are amongst the most expensive medicines in the history of medicine. We're talking about $1 million per dose. And the cost of some of those animal models are remarkably expensive as well. So we need better models for better therapies, ideally. And where are these models coming from? Well, we believe, and this is work that I'm obviously biased because it's work that we've done in my lab for a while, and this is what we focus on, is that we can make use of making muscle from human stem cells to bypass some of those challenges that I mentioned to you before. And particularly from this class of human stem cells called human induced pluripotent stem cells. For those of you who are not familiar with this technology, those were the cells discovered by Shinya Yamanaka, who you see on the right there, who was awarded the Nobel Prize together with John Gurdon for his discovery in 2012. And what Shinya and his team did about 2 decades ago was to do something fairly simple, but it was a genius thing to do. So they took somatic cells, so any cell of our body, skin fibroblasts, some skin cells, for example, they put them in culture, look at many different factors that could be given to those cells to turn them now into stem cells and identify those factors. And now they made super potent, if you wish, stem cells out of very easily sampled cell types, which with the right recipe, with the right method and the right protocol can be differentiated, we say, so can be turned into any cell type we want, including cells relevant for today's talk that I mentioned already to you before, and we can make better models with those cells. And I was lucky enough to be in the right place at the right time, and we were amongst the very first groups to show that actually you can make muscle fibers from human iPS cells, particularly from patients with muscular dystrophy. And you see an example here of our initial work on this now over 10 years ago. And you see in red all the muscle fibers this is the pizza now. Again, we're looking in a monolayer, in a thin layer of cells, with those nuclei scattered around the fibers. Those fibers can twitch. You see the movement if you zoom in closely enough. You see some of them with those striations of the engine of the fiber that I showed you in the movie before. They're twitching. And if you take those muscle-making cells, those stem cells, and you transplant them, you put them in a mouse model of a muscular dystrophy, you'll see that they can generate, now you're expert, you see the myofibers there, with a red rim, which is indicating that they are coming from the transplant, and they even contain a human nucleus, which is green inside. So they can do their job. And we were quite happy that then, following this work and also work done at the same time by other leading labs like the one of Rita Perlingero, many others have followed and tweaked these technologies further down the line. And now, depending on what you need to do, depending on what pizza or lasagna you want to make, you can pick your favorite recipe and make your favorite model out of that. But back to the lasagna, back to 3D now. And 2D. Is it what I showed you now in those images really enough? Is 2D what we can really rely on? You know, we want to be able to study the muscle development and have a large, nice three-dimensional structure which contains all the friends of the muscle fibers so that we can study which one is not working properly. And to tackle this question, really we moved a lot from 2D to 3D and developing this so-called human stem cell derived mini muscles. We were quite good in making muscle from iPS cells, and we took that expertise and combined it with other recipes to make blood vessel cells from the same iPS cells or nerve cells from the same, and combine them all together with special materials called biomaterials. And we made this little 1 centimeter long human iPS derived mini muscles, which we showed were able to model typical features of muscular dystrophy in a dish without having to, for example, look at those features in an animal model. And these are some of the papers we published over the years on that work, and other labs have been following this work or developing other variations on the team, and you will bump into the literature maybe with similar things with different names. So sometimes we refer to them as 3D muscles, other times as muscle organoids or myobundles or engineered muscles. These are sort of different protocols to make a three-dimensional structure that can be used to study muscle features more in detail. And this is what I'm referring to. Now you're looking at the central bits of those mini muscles in this movie. And this is just after 7 days in culture. You see the appearance of those muscle fibers in green, and some of them also start expressing dystrophin. This is a healthy muscle, and again, you know everything about dystrophin now. And what we can do in the lab, and this is work that's been done by a talented senior member of my lab, Valentina Leonello. Valentina set up the conditions in our lab to look at stimulation of those muscles so they can respond, as muscles should do, a generate force, contractile force that is measurable. And I told you it's important also to have, next to the muscle fibers, also the important cell types that are relevant for them to do their job. And here you see an example of a very challenging and rewarding experiment that we did, in which we were able to grow multiple cell types from the same iPS cell. So from the same patient, imagine taking the muscle fibroblast, the skin fibroblasts, the skin cells, turning them into inducible pluripotent stem cells, and from them generating all these cell types you see there: blood vessels, nerves, muscle fibers, and we can see them all together into this staining of a muscle, engineered muscle, a mini muscle, which is oriented diagonally there, if you want to look at them in the right perspective. Now, what can we do with this fancy tool? Now, a few years ago, together with my friend and colleague Pete Zammit at King's College here in London, we decided to use this technology as avatars in a dish, if you wish, of individuals with a specific severe congenital muscular dystrophy called LMNA-related congenital muscular dystrophy, or for brevity, LCMD. This is actually the condition of those images you saw a few slides before in black and white of that child needing respiratory support and feeding support and with severe contractures, to give you an idea of the severity of this condition. It's caused by changes in this gene called LMNA, which produces a protein called lamin C, which lives, you see there in orange, at the edge of the nucleus. Right now we're not dealing with the protein which is at the edge of the muscle fiber, it's at the edge of the nucleus. And if the nucleus contains our genes and the DNA, so the books, as I told you with the Lord of the Rings, imagine this as being the bookcase, right? So this protein makes the shelves where the books are there, and if it's not there properly, then these books can fall apart and the information is difficult to find. And this is what I mean by a malfunctioning nuclear envelope, for those of you who are more expert in those terms. Now, why do we want to do that? Again, this condition is severe, there are no treatments, and it's still a mystery why this causes a muscular dystrophy. This genetic problem causes a muscular dystrophy. And to cut a long story short, work that we've done over the years, and now it's led in the lab by Dan Moore, another talented postdoc, with funding we received from a number of fantastic charities, MD UK most importantly, and then more recently also by QCMD, LifeArc, and AFM Teleton. So what we've been able to show over the years is that if you now look at the shape of those nuclei, the shape of the bookshelf that I said before, in the mini muscles, it is not normal. And you see on the left a healthy looking mini muscle with the nuclei now in red. They are rounded, they are unremarkable to some extent. Look at the right side, you see one mini muscle made from one of the individuals with this condition, and you see these nuclei are severely elongated. They are really, really long and atypical in shape, and you appreciate them from different angles in this movie over here. This is something we can measure in the lab, and we can also see, we saw actually that it correlates with disease severity, sometimes the amount of atypically shaped nuclei. And being a measurable thing, we can use that to develop therapeutics, to screen for treatments in those mini muscles, rather than having to do that in systems which are not optimal to do so. So this is just a little example of what we call complex or advanced disease modeling using some of these new technological tools. But can we really move from disease modeling to therapy modeling to speed up development of treatments? I won't have the time today to tell you what we've been doing and what we are still doing to develop complex therapeutic strategies like tissue replacement or cell therapy, like the one I mentioned to you before. But I want to spend a couple of words on what we're doing to optimize, to improve the development of genetic therapies, particularly around gene therapy. So can we use this mini muscle really as patient avatars to develop safer and more efficacious muscle gene therapy? By gene therapy, I mean, again, the relatively simple concept of hijacking a virus, making it unharmful, and loading the virus a gene, which is now going to be helpful to rescue or to ameliorate a condition. In this particular case, for example, a gene, the healthy copy, the fixed copy of a gene, which would have otherwise caused muscular dystrophy. And why we need to do so? Well, gene therapies are very much into clinical trials in muscular dystrophies and in neuromuscular disorders, but we're still facing the challenge that most of them, which are more often than not this class of viruses called AAV, which are used for this work, adeno-associated viral vectors, most of those viruses have been studied again heavily in animal models. And the understanding of where these viruses will go, and we want them to go very much to the muscles and not to other organs, is coming from studies in animals, which quite often have a different specificity for that process. Something that will go into the muscle of a mouse doesn't necessarily will only go into the muscle of a human, for example. It might go into other tissue and cause side effects. So we want to have a system which makes us look into that and into many other options, and we think that we can harness the power of our mini muscles, if you wish to do so. And to this aim, I had the pleasure to put together this large group of colleagues in this project called MAGIC, which is funded by the European Union, together also with the UKRI and the Swiss SERI agency. And you see that we are a bunch of experts from all over the world, from 10 different countries, all focused around generating new muscle models, which we'll then use for developing specific muscle-targeted genetic treatments, particularly around gene therapies, to answer some of the questions I mentioned to you before. And just in my final slide, I want to give you just a simple flavor of the work we're doing in this consortium. And you see an example here of work that is now then currently led by Somit Dastidar in the lab, another senior member of my team. You see here that we've been essentially giving to the mini muscle one of these engineered viruses to, in this case, produce a fluorescent signal. So if the virus goes into the muscle in the right place, we will get a fluorescent signal there. And you see that we can tell over just the space of about 9 days which of the muscles got a high dose of that and which ones of the mini muscle got very little and which one got none of it, which is something that makes us quite excited. And we're doing a lot of work in this direction to develop new muscle-targeted therapies in humans, if you wish, directly by taking advantage of these systems. And with this, I have finished, and I hopefully managed to convince you that all models are wrong, but some are useful, and hopefully some of them will be useful to help development of treatments for muscular dystrophies over the next years. And with this, again, I've finished my talk, and I really have to thank my lab. I have the pleasure to work with a fantastic group of people, and I didn't have the time today to present all the different projects and the work that they're doing, but you see them all listed there on the left, including some of our previous lab members, which were instrumental to set up some of the systems that I showed you today. Our fantastic collaborators as well, the clinical team at Great Ormond Street Hospital, Pete Zammit, I mentioned already before, Francesco Montoni, Andrea Serio, Maria Mendola, and the entire MAGIC consortium, and also the Crick colleagues over here and the Crick facilities, and our funders, without whom I couldn't really be standing on this fancy stage telling you about this. Obviously, our large government funding is instrumental for that, but it's also extremely important, if vital, I should say, the support we received from the patient advocacy groups and from the charities that I listed here in this slide. Again, a special thanks to Muscular Dystrophy UK as well. It's been funding us for the last 10 years or so, and they're all here today also to answer some of the questions that I won't be able to answer if you have something very clever and difficult for me. Thanks again for your attention.